Well, hello everybody. I'm Christoph from the Department of General Microbiology in the lab of Jörg Stülke at the University of Göttingen. And I'd like to present to you two databases we've recently released, both of which are on the functional annotation of Mycoplasma genomes. So the names are SynWiki and MycoWiki. And while SynWiki, as you might be able to imagine, is on Syn3A, MycoWiki is on the Mycoplasma pneumonia. Both of these databases are already up and running, so you can access them via the URLs down here. But if you just Google those names, you will also find those as an exact search result. So SynWiki and MycoWiki are model organism databases on these two organisms. So that means they host many different kinds of data. So they have functional annotation, nucleic and protein sequences, omics data, structural data, and so on. And for all of these different sorts of information, we have different pages and also interactive tools to kind of explore these data in an intuitive way. Both SynWiki and MycoWiki rely on the same framework. As you will see later, they both have kind of the same basic layout, and also the general structure and organization of the data is the same. And both of them are intended as community resources, which are open for contribution. So people can register and also contribute some data. In turn, SynWiki and MycoWiki they contain a mixture of manually created expert knowledge and also imports from other databases and other relevant publications. So for example, for MycoWiki, a lot of the original data comes from the MyMPN database from the Serrano lab. And as an example, for a recent update of SynWiki, we recently added a lot of characterizations for proteins and enzymes from those two really great recent publications. And now I thought, instead of just showing you some screenshots or anything, I would actually like to show you those databases live in action. So I'm going to switch over to my browser for this, and I hope everybody can see my browser now. And if you do, you will see the front page of SynWiki, where you get the search bar, where you'll be able to look for genes. And we're now going to have a look at the RPOD gene, which is an RNA polymerase sigma factor. When you type that in, you will get redirected to this page. This is the gene page on RPOD. And all of the data in SynWiki and MycoWiki are kind of organized around the gene pages, where you can find most of the data on this particular gene and its products. So as you can see, you get some little description, some data in tabular form. Right below that, you already get some sort of interactive display, which shows the genomic context, where you can also scroll through the gene neighborhood, for example. Further down below on the page, you can find a section which contains all the categories this gene or protein in question is assigned to. So for example, RPOD is assigned to the transcription category. We can click right here, and then we can also see all of the other genes of the same category. And you have this sort of tree-like structure, which has been specifically created for SynWiki to match the purpose best. Right after that, there's a rather recent feature, which is the homolog section. If you click this link right here, you will see a list of potential protein homologs, which are the results of a FASTA alignment analysis. So here you can see, for a fixed selection of organisms which are deemed representative in some way, you can find potential homologs in those organisms with corresponding links, and then also some metrics for identity similarity, and so on. Another feature on the page is here. Well, you will find some other sections in the following, which should be rather self-explanatory, right, in this wiki-style format. Importantly, at the bottom of the page, you can also find some very relevant publications regarding this gene or protein. And you will also find citations throughout the main body of the text. If you have a look at the sidebar, which is on the right side of the page, you will find a selection of links where you can also, for example, export data. But you will also find another recent addition, which is the 3D viewer. There you can see all of the structures which are available for this gene. So it could be RNAs or proteins, but also complexes. So in this case, we have some complex from the protein data bank, but we also added predictions from the AlphaFold database to all of the proteins in SynWiki and MycoWiki. So even if there's no experimental structure available yet, with this you can get some sort of idea of what it might look like. And of course, there's also a link to the AlphaFold database right there. Now I'd like to show you an interactive tool, which is the genome browser, which you can find in the top bar. There you will get also represented with a similar display to that which we have already seen. Here you can also scroll through the genome and click on different genes, and then it will load the corresponding DNA and protein sequence at the bottom of the page. You can perform searches in this sequence, and you can also toggle the reverse complement. On the top of the page, you also get some other advanced search options which might be useful. For example, here you can include upstream and downstream regions, which might be nice for primer design. Okay, and now just to show you how similar SynWiki and MycoWiki are in structure, I'm going to head over to MycoWiki, where I'll type in SIG-A, which is the homolog of RPOD in Mycoplasma pneumonia. And as you can see, aside from the color scheme, maybe a lot of it is really the same. You get the same basic structure. One difference here is the inclusion of some cross-link data in the 3D structure viewer for the AlphaFold predictions from a recent global interaction study from 2020. And from the same interaction study, we also got imported some interactions, which you can see in display right here. For example, MPN 530, which is not yet characterized. You can see it has some interactions with these other sigma factors, for example.
And Christoph, these are experimentally determined interactions as opposed to predicted interactions.
Yeah, that's right. And that's right. Yeah. So there was a cross-link study, and for a certain threshold, they determined this would be interaction. So this is from a cross-linking study, a global analysis. If you want to have a better look on those interactions, you can also use the interaction browser, which you can find in the top bar as well. There you have some sort of dynamic display where you can also click on genes and it will give you a summary, but you can also increase the radius of this network. So instead of just including the immediate interaction partners, it will show you the partners of those, and you can expand the network this way. And then you can see if you expand it far enough, you will find that there will be some clusters you can observe, for example. Another useful interactive browser is this expression browser right here, which features omics data from a 2011 study. And there you can see the expression, or here in this case, the protein levels for SIG-A under different omics condition. You can also click on these data points here, and you will see the corresponding description. And additionally, you can also compare different transcript levels or protein levels. So here, you can compare it with NPM 530, and you can see the different expression levels under those conditions. Okay, so much for the browser. I hope I could give you a rough idea of what it looks like and how you may be able to use it. I'm gonna head over to the presentation now. And yeah, just to give you a little outlook of things we want to implement in the future. So there's this flags tool, which allows you to compare gene neighborhoods across different genomes, which can be helpful in the characterization of some genes. And we plan to implement that in our genomic context viewer. And another thing we'd like to do in the interaction browser, currently you can only see protein-protein interactions, but we would also like to include RNA, metabolites, and complex into this mix of interactions in the future. If you're planning to use these databases in the future for your own research, please use our most recent publications. You can find citations for these also on the pages themselves. They're linked there. And there's one thing I'd like to ask of you. If you're in any way interested in contributing to these databases, that would make a huge impact. The more people contribute to these open resources, the more utility they gain for the research community. So we would really appreciate that. If you're interested, please just contact us. You can use this email address. And yeah, we'd be happy to create an account for you. So with that, I'd just like to say thank you for your attention and for this opportunity to present these databases here. A lot of thanks goes to Thiago Pereira, who was working on those resources before I joined this team at the end of last year. And of course, the current wiki team as well, which me and Vincent's working on the technical side of things more, and you're being responsible for a lot of the manual creation of data. So thank you very much.
I'm continually impressed. You realize that this makes the sixth, you will now be the sixth group that has made a computational prediction of the genes in the minimal cell, starting with Antoine Danchin in 2016. And it would be, so I'm going to put together a table in a paper that will be coming out of the JCDI and Illinois groups that has a table that compares the annotations of all six databases. But again, you have the advantage of AlphaFold and interactome data that other folks didn't have. When will your interactome studies with this organism be likely published?
Maybe I can jump in here. So I'm
sure, Jörg.
Yes. So I really cannot say so far because this is still with our colleagues in Berlin. And for Mycoplasma pneumonia, I think it took 7 years or so from the initial data collection until publication. So I have to ask you for a bit of patience.
Sure. Sure. I will be writing you, Christoph, to ask for editing capacity, if that's okay. And probably some of my friends, some other groups who are listening today.
Yes, if I also may add something. We have recently had a conference on Bacillus subtilis, one online and one also in person. At both opportunities, Christoph has done a SynWiki workshop, so which showed the features of the database, but also how to edit. And I think if people would be interested in adding, we would be very happy about this. It would be a nice idea to organize such a workshop of about 45 minutes to an hour for those who are interested, to show you how you can do it. It's really very, very easy, but I think it's good to have some introduction how to do it.
I would certainly show up. Another question. I noticed that you were comparing to Mycoplasma mycoides subspecies mycoides. So I assume that that's the Anya-Person 2004 genome. I don't know.
I assume so. This FASTA alignment analysis was done by my by Thiago Pereira, and I assume he relies on that. But I'm not sure. I'm sorry.
It would probably be good when you have that you designate which strain that you are using, because some folks might not, there are those who use M. pneumoniae Eaton and those that use M. pneumoniae M129 and similar things elsewhere.
Right. Yeah, true. That's I'm gonna try to include this data. Yeah, good point. Thank you.
Other questions for Christoph or Jörg? So it does seem, though, that you're gradually putting a few interactome data out on SynWiki.
Yes, indeed. That's all a like continuous integration of data based on manual creation. Yeah.
Okay.
Exactly. And the more people contribute to this, of course, the more encompassing it will be. So yeah, I would like to again invite you if you're interested in contribution.
You'll be hearing from me really soon, and hopefully other folks who are listening today.
That would be great. Yeah, thank you.